into our next big unit. So I'm going to give you a hot second to do that. Hi, and Jasmine's going to restart the recording, so if you guys just want to view the introductory material or want to start on the biomolecules material, there'll be a clean cut for you to find. All right, got me going? All right. Okay, so our next unit is going to be focused on biological molecules. Remember when we first started talking about this, I said we're really going to work on building our themes or our content from the smallest to the largest. And that's really what we're going to be doing here. So when we say biological molecules, we're really talking about what are the basic molecules that we need to create life, or what does life need to sustain itself. So those are the types of molecules that we're going to look at. What are they? What are they used for? And how can we identify them? So here's what we're going to do as we go through this. So you're going to see these types of roadmaps a lot. They'll be really valuable to us. And this is how many of our units are going to look from here on out. So much of the time, we're going to start with some important vocabulary that's going to help us navigate whatever unit is to come. And then we'll have whatever the major pieces of that unit is. So in this case, you see we have our 4 different major biomolecules that I want to touch on. So in this case, we have carbs, lipids, proteins, our favorite biomolecule, and nucleic acids. And then you see here on the right-hand side, I have a helper box. That's the right. Okay. And in the helper box, I have, how can we focus on this material a little bit better? All right, so how can we make sure we don't get overwhelmed? So as we go through this material, I have, what should we be looking for? Okay. And this will stay up every time we revisit our roadmap. So we are reminded what is the most important information that we want to be looking at as we go through our 4 biomolecules. And this will help us. You'll see the same reflection on the goals listed on the top of our e-learn page. I am not breathing in between here. What? Okay, vibe check. That actually happened, right? I didn't touch anything, man. Shouldn't happen. We're just going to keep going. It's fine. If it happens again, I'll worry about it.
Dr. Annette, you're no longer sharing your screen on Flash.
You're kidding. Why? I didn't do anything. Okay. Today's pause while we deal with some minor difficulties. Why? Okay. All right. We're just going to sweet talk it. It's just got to last another like 30 minutes. So that's someone else's problem. I've totally lost my place. The things will be here to guide us on what we're doing. Okay. I'm all like, I lost my mojo now. Okay, so let's start by looking at some important vocabulary that's going to help us deal with our biomolecules. And specifically, how do they exist? How do we create them? What are they? And how do we break them back down? Right? Because ultimately, when we're talking about biomolecules, we're talking about building blocks, we're talking about food, right? How do we have resources? Okay, so term number 1, monomer. Okay, some of these may be more familiar than others. So when we use the term monomer, and specifically we're going to focus on some of these prefixes because they're going to help us navigate through a lot of terms. All right, we know mono. I'm drawing here. Okay, means one. So this is going to be anything that we encounter that is a single version of that biomolecule. So it's going to be a single chain or ring. So an example of this that we're going to encounter right away is a monosaccharide. Okay. We're going to see this right away in our carbs. A monosaccharide is just a single sugar. So the saccharide ending, we'll see this in a hot second, is sugar or carb. I just have an example of that right in the picture below. Right, we see single or one, so we have one loop or one ring. And we'll actually get to deal with this sugar in lab in the next week or two, depending on which group you are. So this is one of the simplest sugars that there is, glucose. Okay, so when we say simple sugar, we're literally just referring to its structure. The fact that it is a monomer or just has one ring. So it's small. Not so bad. Okay, let's compare that to polymer. Okay, so in this case, polymer, we have poly. So instead of mono, meaning one, we have poly, which means many. Counting in science gets real easy. Okay, so my example here, instead of a monosaccharide, we have a polysaccharide. Okay, just meaning that it has lots of rings. Right. So we're going to see this come up a lot. Okay, so you see here specifically we have a disaccharide sitting out. Okay. Counting in biology is super easy. We have mono. Woo, 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 woo. My spelling needs help. We're cool. One. Di. Two. Poly. That's it. Everybody learned how to count today. Okay, so we lazy creatures, because nobody cares. Okay, so mono just means one. These are simples. So you can see I have my di being two rings, or more than one, right? So here's my disaccharide in the corner. You guys will get to play with this as well in the coming weeks. One ring, two rings. Okay, slightly more complex. Okay, so this is sucrose. Sucrose is functionally table sugar. Okay, so the stuff you put on your Cheerios. Okay, and then lastly, we have our polysaccharides. Okay, polymer means many, so literally 3 or more. And or more can mean anything. So I have 3, I have 4, I have 5, I have 500. Does not matter. I do not care. Just many. All right. So in this case, all of these do have more than 3. This is just how lazy we are. So I have 1 ring, 2 rings. Good gravy. 3 rings, Okay, and this could repeat for infinity for all it matters. Okay, but it's 3 or more rings. So we know it's poly. Okay, so these are starches, kind of like what you find in a potato or cornstarch. The bottom one here, which is very similar looking, is cellulose. Like that's the structural component you find inside plants. Makes them stand upright like little flowers. Okay. But the key here when we're looking at this is we're just counting. All I care right now is we're learning how to use the terms for counting. Okay, so mono being 1, di being 2, gotta end in a no now, right? Poly just meaning more than 2. So these terms aren't too bad, right? Counting feels very relaxed. It's a nice Friday activity. Okay, any questions so far? Okay, and we'll use these saccharide terms a lot once we get into carbohydrates. So if those feel a little stressful, that's okay. We're going to get a lot of practice with those. And how we can put together and take apart biological molecules. And so we were interrupted a little bit with this. And so the first thing we learned was hydrolysis. And so remember, as we looked at hydrolysis, our goal was to break up Janet and Carl. So remember, we wanted to look at the word hydrolysis. In particular, we focused on lysis. This is a word we're going to see a lot. Lysis means to break or take apart. And of course, hydro means water. All right, so we're using water to break Janet and Carl up. Remember, we saw that we had Janet and Carl, and this is our couple, Janet and Carl. All right, and so here we have a dimer. Okay, so remember, di means 2, or a disaccharide in this particular case. Okay, so we have Janet and Carl, and remember they're being held together by their handhold. And that was our big key, is we wanted to focus on breaking that handhold, that oxygen, right? We kind of thought of it as a fist being held together by 2 arms. And so remember when we went through this? Oops, use that color. Okay, remember our first step is we're going to break one of their arms, right? We need to break that. Sorry, Carl. All right, so someone's got the stumpy elbow, okay? Someone else is going to get the fist, right? No breakup is equal. It's the reality, okay? And we're going to use H2O to fix this so they can both be whole individuals going about their single life. Okay, so remember when we look at H2O, we have an oxygen and 2 hydrogens. So we're just going to distribute those to make sure everybody has hands and gloves. Okay, that was our big goal here. Do this from scratch. I like doing it from scratch. Keep Janet and Carl on the same side. Okay, so Janet, remember, is easier because she kept the fist. And keep more than that in the divorce settlement, which we will do later. All right, so she already has an arm, so we're going to keep her arm and her 1 oxygen, so she kept the fist. So all she needs is a mitten, right? So we're going to give her a mitten, 1 of the hydrogens. Cross that off the list of things we've used. Okay, Carl. Okay. He only has an elbow, remember, because of where we broke this. So Carl needs a fist. We're going to go ahead and give him a fist. Cross off the fist. Okay, and then Carl also needs a mitten for his fist. So we gave him the mitten too. Oops. Okay. So when we do that, right, we used all of our H2O. Janet and Carl, both completely single, ready to mingle. Okay, and whole individuals again. Okay, and both were monomers. And this was the big key, right? We took one big biological molecule and made two little ones. And this is fundamentally where we ended class because things kind of went hot in the handbasket real quick after this. Okay, this feel a bit familiar? Scraping off some of that weekend dust. Okay, so our goal at the end of class that we never successfully managed was to compare this to dehydration synthesis. Okay, so the big goal here is we're going to invert the process. So where hydrolysis, the goal was to break up Janet and Carl, dehydration synthesis is all about putting them together. Okay, so remember we looked at how can we tell this from what the words are saying? So we have synthesis, right? And so the terminology for synthesis, hey, meaning to make, to synthesize, right, to create. Okay, and we also know dehydration, okay? We're going to pull water out. We're taking that away. So we're pulling that water away instead of putting that in like we did before. Right, so we're taking away those mittens. And the other way that I like to look at this or try to remember this is for our hydrolysis, right? Hydrolysis was one word. Ooh, just rude. So we have one word. Okay, and we're starting with one molecule. Okay. So hydrolysis is one word, starting with one molecule, and our goal is to make two molecules. Right, so hydrolysis is one word, we're going to make small, right? One word, one molecule. Start big, make little. Dehydration synthesis is the opposite, right? We are two words, and so we're going to start with, what's showing up? Two molecules, right? Because this is additive. So we're going to start with two smaller molecules in order to make one big one. So two words, two molecules. So how does dehydration synthesis work? So let's bring back Janet and Carl. Yo. Okay, and now they're single. So they look like the end, right, of our last process. And now we know our goal is to make that handhold. So we're going to look down here at the hands we have. Okay, we know our goal is dehydration. So I need to pull out water. Okay, and so we remember water is very simply OH. Okay, so that's what I need to take away. Okay, so first thing I'm going to do, start simple. Let's take some mittens. So I know that my O's, we've been talking about these being my hands. So here are my two hands. Found some hands. All right, O's look like hands. So I found some hands. So I'm going to pull the mittens off of the hands first. That's easy to spot. It's easier for me to find. So here's a mitten and here's a mitten on the hand. I'm going to take that off first. Okay, so we have 1, 2 hydrogens. Okay, 2/3 of the way there. Okay, now I know I need an oxygen. So I need one hand. So I'm going to look at Janet and Carla, and I know I need to take one of these hands. Now, it doesn't matter which one, right? I just need to take one of them. So I'm going to pick on Carl because we picked on Carl before, so we're just going to pick on him again. So here's an oxygen. I'm going to take that oxygen. Check. Okay, so by taking 2 hydrogens and an oxygen, or 2 mittens and a hand, okay, I have dehydrated, right? I've taken out a whole water molecule. Okay, so that's where this is coming from. Okay, that's my H2O. Everybody see that? Okay, so what I'm left with now is Janet has one fist. She's got one hand left on this side. Still single, right? And Carl, okay, who has no hand on this side, right? Just the elbow. Okay, so they're primed and perfect, right, to hold hands now on hand and elbow. Okay, so here's Janet's arm and Janet's original hand, which is going to combine with Carl's elbow. So now they're holding hands over the leftover hand. So it really is just the inverse direction from what we did in hydrolysis. And so the key here is because we took the water out, okay, they're going to hold hands, and if we push the water in, right, we break their handhold. So this is a really good thing to practice, right? Dehydration synthesis, making hold hands, and hydrolysis, breaking handholds. Okay? So I have a good practice exercise, no points worth, right? So it's just letting you get practice if you want to on our homepage, but let you work with Janet and Carl and some others. I make like a thruple, so you can make them really big and really small just to get practice on working with dehydration synthesis and hydrolysis. So if this isn't feeling very solid, we're going to practice it a little more together still, but it's a really good tool that you can use. Okay. Any questions on what we've done together here and the difference between hydrolysis and dehydration synthesis? Okay. The tool worksheet that I've given you then is good practice, and it's also something that's good to bring to a tutoring session or to one of Jasmine's TA sessions as well to ask questions about. Okay. So any questions about any of the vocab, the general vocab we went through before we dive into our 4 biomolecules, starting with carbs? Gracious. Okay. So we're going to start walking through our 4 biomolecules. So on my roadmap back up here, you see I have my help box. So remember, as we go through here, the big key is how can we stay goal-oriented? So when we do this, right, for each, okay, I really want to stay focused on what do these biomolecules do, right? What are they used for? Okay, and then, okay, how would I recognize this biomolecule? Okay, so what makes it iconic about its structure? Okay, this will be a little harder at first because we'll only have the one. But eventually, right, we'll have several, so you'll be able to say, how would I compare them? How would I tell them apart now that I've seen a couple? Okay, so let's go ahead and start. We're going to keep my orange box here in the back of our mind, okay, as we go through carbohydrates. Okay, so carbs. These are one of our more familiar biomolecules. Serving largely as fuel, right? This is a food item, as well as building materials, and we'll see them as both. So when we talk about these, this is fundamentally sugar, okay? And quite literally sugar as you picture it, but also forms of sugar as they go in increasing complexity, right? So table sugar, we can think of complex carbs, right? Breads, okay? As well as when we're talking about building materials, we're talking about things like starches, right? Potatoes, okay? Cellulose, the building blocks of plants. Chitin, okay? The shells on insects. Okay? All of these things are carbohydrates and their variations on. And so carbs, aside from being delicious, are very useful. And is a default form of fuel for your cells. And DoorDash. And so how can we recognize our carbohydrates? So remember I said we would use the word saccharide a lot. Well, here we see it. Okay. The dreaded double C. Okay, so saccharide is just a fancy word that literally means sugar. So this word is associated with carbohydrates because carbohydrates are sugars. So when I say monosaccharides, okay, I'm saying this is a single sugar. Okay, so this form is one of the easier forms to recognize, right? Because single sugars are simple sugars, right? Simple meaning single or small, not easy, right? So if I look at single or simple sugars, what are some of the things that I can start to look for? Well, the first thing I can recognize is what's called a hydroxyl group. That's this top image here. Let's switch colors. A hydroxyl group is also a simple, right, being small, a simple structure. Okay, so this is our hand and mitten structure. So we've been playing with this for a while now, and now we're going to put a name to it. Hydroxyl. So as we can see, our carbohydrates, all of them do, okay, but the monosaccharides most clearly have a lot of hydroxyl groups. So if I pop over to my structure pictures, I can start to circle and see just how many hydroxides I have. Okay, so in the ring, I can see them. I can also see them, oops, I'm sorry, in the chain I can see them. I can also see them in the ring. So the key here is per size, okay, I have quite a lot of these. Okay, so in both of these I have 5 Okay, and given how small these structures are, that's quite substantial. Right? I seem to be circling a lot of space on these structures. And this is really important. Okay? As we're going to find in general, okay, oxygen, which is the core component of these hydroxyl groups, is a really important element. So hydroxyls are very important compounds. Okay, the key is because hydroxyl groups, or oxygens, anything with a lot of oxygen, is what's called polar. Polar organisms, elements, probably too small to be going organisms here. Okay. Things with a lot of oxygen, okay, are also hydrophilic. So when you're polar, this is referring to our oxygen here. Oxygen's like a crappy roommate. Has a lot to do with bonding. So polar things, the oxygen here, okay, crappy roommate. Everyone's gonna have the crappy roommate, right? Everybody buys toilet paper, okay, but then the roommate like uses a lot of it, okay, like way more than anybody really needs to, right? You buy the whole package, you use like 2 rolls, they use the other 8, okay? But I'm only gonna buy it half the time, right? I use it just as much as my husband's like this. I'm gonna use like the whole freaking roll, dude. Chill, okay? Crappy roommate, okay? Definitely buys half the groceries, but eats like 90% of them. Crappy roommate. Okay, this is what oxygen is like. But it's like that with electrons. So with our oxygen, right, we're supposed to share electrons, but does not share. It's like toilet paper. I'm going to use like way more. So I steal electrons way more often, which means I'm going to be like fake negative. Just a little bit. I hog them, hoard them. Which also means like I'm going to interact with things a little more frequently. We do not need to go super nitty gritty here. Okay, but all that means is I like hang out. I do things. Okay, I'm interactive. Okay, so this is what we mean when we say polar. I interact with other compounds. I play well with others. Polar things play nice. Okay, so one of the things I play nice with means I'm hydrophilic. So hydro, we've seen that prefix before. What does that mean? Water. Okay, and philic means loving. I'm water loving. I play well with water. So this is sort of intuitive to us though. So remember that carbohydrates is a sugar. What happens when you put sugar in water? Dissolves. Okay, so this is what we mean when we're talking about hydrophilic. These are compounds that will mix with water. And they mix really well with water because it has all those oxygens, because they're polar. Interacts with other compounds. So we're hydrophilic because we have polar hydroxyls, and we don't just have 1 or 2, right? We have a lot of polar compounds. So when you take your sugar, you pour it in your water, it dissolves. Plays well with others. Polar plays well with others. So oxygen in this case is something in general that we can look for. Do you have a lot of oxygen? Okay, well, you're polar. You with me so far? I feel okay? Polar is a big term. And this is something we're going to use a lot of, so we want to feel good about this. Okay, the other thing, I'm going to change colors here, that we can use to recognize a carb is what's called a carbonyl group. Okay, this is significantly easier to see if I'm in the chain version. Okay, so this looks like this. Okay, we're going to see this R business a lot. R is a really lazy chemistry thing that just means something or anything else. Okay, the thing I'm not interested in. It's the other. So when I'm focusing on the carbonyl, all that is the carbonyl is the C, double bonded O, H. That's a carbonyl. And the R here just means, and then you stick it on something. So if I'm looking at my chain structure, I'm also going to circle my carbonyl in green. There's my carbonyl. C, or carbon, double bonded to an oxygen on one side with a hydrogen on the other. Okay, so it's fairly easy to pick out on my chain structure. Okay. Trouble is in the ring structure, which is the one on the right, we can't really see that anymore. Okay. And that's because of this last bullet, that we get rings in the aqueous. Aqueous means water. Okay. So it changes its shape a little bit when we dump it in the water. So instead of looking like the picture on the left, we get the picture on the right. Fundamentally still very similar, right? We see all those hydroxyl groups and all of that, but we lose a little bit of our main piece. Okay, it's still here. It just looks kind of funny. It's much harder to recognize. This is why for each of these, I'm always going to try to give you a couple of things to look for. That way you always can recognize what we're looking at, kind of no matter what you're given. So the carbonyl is nice, okay, but we also have the hydroxyl groups. Okay, so if I were to say, is this a carbohydrate? We both have the red and the green, okay, our hydroxyl groups and the carbonyl group to look for if I wanted to identify it. Okay, so we have two tools in our toolbox now. Okay, and this is a lot of terms, so we really want to practice this, right? So we now have monosaccharide, hydroxyl, and carbonyl. Okay, any questions? Okay, so remember then, we're going to be practicing our terms. So here's that saccharide ending. Saccharide still meaning sugar. So monosaccharide is a single sugar. Right? A disaccharide is a double sugar. So remember, mono means one, di means two. Okay, so the process I have up here, is this hydrolysis or dehydration synthesis? Good. Right, I have two, 1, 2 that I'm starting with. Right, I'm dehydrating. I've taken water out to create one bigger molecule. Okay, so again, if we would picture this as Janet and Carl, okay, the key here is This is going to happen at our, now we know the term, our hydroxyl groups. So I'm going to find a hydroxyl group on Janet and a hydroxyl group on Carl. I know I need to pull off that water. Okay, so I'm going to pull off 2 hydrogens first. The mittens. There goes Janet's mitten. There goes Carl's mitten. There's my 2 hydrogens. Okay, I still need to pull off an oxygen, a hand. Okay, sorry, Carl. So I'm going to pull off Carl's hand. And there's the oxygen. Okay, so there are dehydrated oxygen. We've done that all from our newfound hydroxyl groups. What's that leave me? 1 oxygen remaining, right? And that 1 oxygen remaining is the hand that they're going to hold on after dehydration synthesis is over. Hi, this seems like a nice drama-free place to stop for today. We'll make disaccharide our teacher question.